Paul Kastner, who's the Director of Research and Target Identification and Validation at Amgen. There's Caroline Breitbach, Director of Clinical and Translational Research at Generex Biotherapeutics. Stephanie Scheffler, Scientist in Proteomics and Target Discovery. And Sylvia Dar, who's an architect and lab planner for Vlad Architects. So before we get into the question and answer period, maybe we could, Paul, we'll start with you since you have a microphone and you can kind of give a brief background about yourself.
Sure. My name is Paul Kastner. I'll give a very brief history of how I got to where I am. I loved math and science when I was in high school, much like many of you do. Thought I was going to be a mathematician first, but then I took a chemistry exam, got a scholarship to go to the University of Illinois in the chemistry department. So I went there, found out that I really didn't want to be a chemist. At the time I didn't see any use for it, which is strange now that I'm in drug discovery. My other great passion was the outdoors. I loved the environment and animals. So I was standing in line to change my major from chemistry to ecology, ethology, and evolution to study animal behavior. And I thought, what am I going to do with that? So I'm standing there and it's next, okay, what do you want to change your major to? And I said, genetics. Didn't really know where that came from, but I became a genetics major. And one of the courses I took was an immunology course. Had a professor, Rod McLeod, that was just so inspiring. Just fascinating topic, and I just fell in love with it. And I went to Harvard to get my PhD in immunology after that. Went on to do a postdoc in neuroscience at UCSD, so switched again. And then went on to a number of little startups. As Susan says, most of them fail. But I worked on antibody phage display, worked on bacteriophage gene therapy. I worked on protein microarrays at 2 different companies, and then found myself doing assay development and high throughput screening for a company called Telluric, which was then acquired by Amgen. So 4 companies in, finally hit one that was successful. And now I do target discovery and validation for Amgen. And love it. It's great. It's perfect for me, a mixture of basic science and applied science, since we're trying to find those targets to treat with our drugs to affect diseases.
Well, I'm Sylvia, and when I was young, I really loved biology and animals and the environment, and I thought I was going to be a biologist. And I also really enjoyed drawing and art and design. And when I went to college, I took classes in art and architecture and classes in biology and chemistry, and I ended up trying to decide whether to major in biology and chemistry or whether to major in architecture. And I decided, okay, I'm going to be a scientist. So I went through the whole path, became an assistant professor of biochemistry, but always in the back of my mind I was thinking about design and architecture. And so at 37, I decided I'm going to be an architect. So I went back to school, resigned my faculty position, went back to school, became an architect, and then when I graduated from architecture school, I thought, I know more about labs than any of these other architects, and I'm going to become a laboratory designer. And so I think what I'm talking about is alternate careers in science. I know it's easy to, when you're just starting out as a scientist, you think, the straight path, become a researcher, become an academic, become a teacher is the only way you can go. But there are many ways that you can take a science education and understanding of science and do other things in the world as well. And in fact, I was one of the designers for Tellaric's first building.
Hi there, I'm Carolyn. I am a Canadian. I grew up in Canada and attended McGill University studying microbiology and immunology, just because I thought bacteria and viruses sounded pretty cool and how they interact with the immune system sounded pretty cool as well. And after receiving my honors degree, I actually went to Vienna, to where my mother was from, and just spent a year there doing, being exposed to arts and taking a couple of business courses. And then I did want to pursue a career in science though, so I returned to Canada and chose a PhD program in a field of oncolytic virotherapy. This is actually using virus and applying that science, using viruses as a therapeutic for cancer. So these viruses actually infect cancer cells but not normal cells. And I think that gets at what Susan says earlier. I really found a passion in that. I decided I was searching for postdoc positions. I was actually accepted to a postdoc at Harvard, but ultimately I was offered a position at a small biotech company here in San Francisco that actually applied these viruses. And so that's what I do now. I design clinical trials. I analyze data from patients actually that were treated with our viruses, and hear those patient stories as well of patients alive over 5 years who had very advanced cancers. So that's certainly also, I kind of listened to my gut. I followed my passion, and I've been very happy. So.
My name is Steffi. I'm a chemist. I studied chemistry in Hanover in Germany, and I also got my PhD there in the Institute for Technical Chemistry. Doesn't sound like biotechnology or something, but what they were doing was biotechnology. And so I came, just in my master's thesis, I came in the field of biotechnology and did tissue engineering and cell culture. And what I did during my PhD thesis was the development of a ceramic material for bone tissue engineering. That means to have a scaffold material for large bone defects. After my PhD thesis, I came to a small company, also in Hanover in Germany, and they were working in the field of in vitro toxicology, mainly for airborne substances. So what I did was I was working with human epithelial cells and exposed these cells to cigarette smoke and to see what happened with the cells that they're exposed to cigarette smoke. Afterwards, I came in May last year. I came here in the US, and since July, I'm working for a small company, a startup in Palo Alto, which is called Target Discovery, and I'm working now in the proteomic research. So we are looking for biomarkers, for protein biomarkers, which can be useful to have an insight into a disease like cancer, for example.
I'm Alex. I took the chemistry job away from him. I was originally pre-med as a high school student, and went into chemistry at the suggestion of our family physician and friend. He was convinced that that a chemistry major would nail the MCATs better than a biology major, and so that was his suggestion to me. And so I went to University of Illinois in Urbana-Champaign and started doing chemistry as a major. And there was a young assistant professor who ended up becoming pretty famous in his field, and I just really enjoyed the class I took with him, so I asked to do research with him. And I got to work with this guy and ended up on a couple of really high-profile publications. And that really, after that, I was pretty much hooked. So I ended up getting my PhD at Harvard, and then did a couple of postdocs, one at Caltech in inorganic chemistry, where I did lanthanide chemistry, group 3 chemistry, and then I moved to MIT for a year and did a little bit of palladium chemistry. And then I started at Cytokinetics, and I've been there now for just now 10 years. And I've worked on several different projects, but I've been super lucky my whole career. My first project is a heart failure drug that's partnered with Amgen, that's currently in phase 2. And the second project I was on is an ALS drug that's also in phase 2.
Well, thanks, Chair and panel, for your backgrounds. I'm wondering, before I throw it out to the floor to get random questions, I think that one of the things that came through to, I think, all of us was Susan's accounting for some of the key people and experiences in her career. And I'm wondering if just briefly, if you could comment on a mentor, the passion, and the aha moment, and what engaged you to go over obstacles that were emerging as you were trying to pursue your career. Because I think that the students here, not having gotten very far, may be interested in hearing these personal touches, because there are people in our professional lives as well as personal lives that pull us through and serve as a role model to tug us along our pathway. So maybe we'll start and go back the other direction.
Well, I always enjoyed doing stuff in the lab for classes and things like that, and I always enjoyed science, but really the aha moment was getting into a research lab. And you're not just discovering something for yourself. You think about it, this is the first time anybody made this molecule, and you're holding it in the entire world supply in your hand. And this little bit of knowledge is you're the only person with that bit of knowledge at that moment. And, so discovering something for the first time, making something for yourself. I mean, I like to cook, but oh wow, another cupcake, whoop-de-doo, right? But you make a molecule, and you can, and then you make a lot of it. You got a jar of it. Those are the things that are really exciting in the lab, and to discover something for the first time, or those are the things. But the thing that really more recent, but you still get them as you go on. Most recently, the drug that's partnered with Amgen in phase 2 for heart failure. You see this molecule that you worked on before and after with this heart echo, and you see it pumping better, and you're like, wow, that's a little teeny tiny molecule, molecular weight 400, that's doing that, and that's really cool. So you can have more than one of those times in your life, and you'll see that over and over. It's not just a one-time thing. You'll have multiple ones like that. That's one of the exciting things about being a scientist.
I think for me it was a little bit like for Susan. So my parents are both chemists, and I think so science was always around. So I didn't know that I want to be a chemist since, I don't know, maybe the last 2 years in school or something. But yeah, science was every time around, and I did my internships during school in different labs and tried to see different things in the lab. And so I always knew that I want to be a scientist, but then, yeah, I decided very late that I want to be a chemist. Honestly, I don't know why. So it was just like, okay, maybe I'm a chemist, I don't know. So, and now I came back to the biochemistry or biotechnology because it felt for me like being, I don't know, maybe supportive for people and that you can really help and do something. So the biotechnology was, yeah, closer to the target for me. So that was the reason why I went to the biochemistry or the biotechnology than from like something like organic chemistry or so.
So I do have those aha moments that also stand out, and it should be said that of course they are few and far between. You're going to have 99 failures for the one success, but it's worth it. And I do remember that feeling when I was at the computer where that result came out, and I just remember thinking, I know this is going to be a big story, and right now I'm the only person in the world that knows this right now. So that definitely is really exciting, and even though now I don't work in the lab anymore, again I work mainly on patient data, well, I still derive a lot of satisfaction from my job because I'm still exposed to that primary data, and that's still quite important to me, even though I like that it's applied. In terms of mentors, I certainly think to my PhD supervisor, Dr. John Bell. He actually lost his 2-year-old daughter to cancer, and so he has a very personal connection to our field. And again, he is a PhD scientist. He has a virology background, but was very unusual in the academic science world, was wanting to have his research be very applied, and that's why he actually co-founded the company I work for now. So I think because of he's of course a rigorous scientist and very well known in this field and publishes well, but I think he brought a unique passion to the field that really affected everybody that worked for him. Even if you had a preliminary result, he would be genuinely excited, and that would make you want to get back into the lab and confirm it and expand on it. But he certainly was a big mentor for me and really listened to me in terms of what I wanted to do career-wise. Again, I'd actually committed to a postdoc already at Harvard and kind of thought I was going to go that regular career path. And then he said, you know, you mentioned that you wanted to do applied science, you cared about science and patients. So I'm going to tell you about this position for you in a small biotech company. There are 5 people working at Genrex when I joined. So I really appreciated that he also listened to me. He thought, I wasn't sure if I was going to tell you about this, but you're a big girl, he said, so you can make the decision yourself. And I ended up making that decision on quite short notice. To take that alternate career path because again I was interested in the applied field. So
unlike many people here, my parents were not scientists, and I was the first person to go to college from my family. So I think that my passion for science kind of came from, I don't know, from inside me. I remember the first time I learned about Mendel. And I was, I don't know, 12 or something, and my mother kept saying to me, why are you growing pea plants? You know, because I turned the garden into some pea plants that I could try to make those peas change color like Mendel did. My first mentor in science was actually my academic advisor in college. You know, I was a biology major, and I was sitting in his office talking to him, and his office had a door that led to the lab, and I said to him, what are you doing there? And, you know, of course, he seized that moment, was happy to have more people working in his lab, and that was the beginning of my life in research. I have to say that some of the things I enjoyed best about science, or I enjoyed about science, was solving problems, like Susan, trying to find the answer to that question. And for me, many times, the biochemistry work that I was doing came down to a spatial question. What's the structure of the enzyme? How's it interacting with the, you know, with whatever molecule it's interacting with? And so making the change to architecture was, in a way, just taking study of structure to a bigger level. And you know, knowing who the people are who are affected by your research is very powerful. And when you're an architect, of course, the people you're working for are sitting right across the table from you. And it's easy to see that you're doing something that is beneficial to them. And also, in my case, my mother understands architecture much better than she understood biochemistry.
So my father is an inorganic chemist, a professor of chemistry. My mom was a nurse, but neither of them were really mentors. They never pushed me down the scientific path. But I'm very lucky to have a lot of different mentors along my path. I remember one of my summer jobs was at Rush University, worked for Dr. Eric Bramer. And I was purifying a ganglioside from cow brains, you know, not very romantic. You put a brain in a blender and extract it. But it was a great job because I could sit down with him and, you know, ask him, you know, why am I doing this? And, you know, he launches into, you know, a long description of the work and what he's trying to do. As Susan said, you know, if people are interested in you and you're interested in their work, take advantage of that. Start asking questions. You'll learn a lot, and you never know where you're going to get inspired. So Eric Bramer, the professor, Rod McLeod, that I met at U of I, who inspired me to go into immunology, he was the same way, just so passionate about the field of immunology, the study of immunology, that he spurred me down that path. My graduate advisor, Martin Hemmler, same thing. He just loved talking about the science and the pursuit of knowledge. I could go on and on. When you find those people, take advantage of them.
Thanks a lot. Thanks a lot for these stories. One of the things that strikes me as I listen to the stories of my colleagues up here and Susan's and reflect on my own is the combination of passion together with scientific inquiry. And I think a lot of us, maybe on the stage where in our high school years, probably people would point to us and say, well, there goes nerd number whatever. And what I think this exemplifies in this small little meeting here is that people that have scientific interest and desires are just as much as people that don't have our particular desires. And so just because your passion lies along a vector that makes you unique, I think it's really nothing to be ashamed of and something to really actually leverage for the future. So I think that from this discussion, I've sort of extracted that out of this. But I want to now throw our panelists and myself open to your mercy, really, because it's the young who can ask the most embarrassing questions of the old. And so we're up here sort of waiting to be embarrassed or called on the carpet by you. So don't be afraid. This is a very informal, although it feels formal, but it's a very informal meeting.
What are the differences between working in an academic research lab and an industry?
I'll go ahead and repeat the question, so in case people didn't hear. The question is, what's the difference between an academic setting and an industrial setting? I don't know if anybody would care to take that on.
I'll start since I have the microphone again, and then I'm going to hand it off. I'd say probably the main difference is that in an academic lab, you can really follow the research. Take advantage of the observations that you're making from an experiment that you've conducted. It might not answer the exact question you had originally intended, but it will show you something new, and you might have the ability to follow that in academia. In an industrial setting, we really need to be focused and applied. We have our eyes on an end goal, and we have to get there, and those interesting observations, we might have to just file in the back of our brain for now and leave it aside and keep going to where we're targeting. But I'll let anyone else.
I think the biggest difference is when you're in graduate school, a focus is on performing experiments and having a body of work that you can call your own, and you have to basically push that aside in industry because you can't do everything yourself. There's multiple disciplines. There's more work within your discipline. You can't do it all. So the biggest, his differences are certainly there, but the other difference you'd really need to focus on is being part of a team as opposed to just being somebody who's just cranking out experiments, because the team aspect of it is a big deal, and you can mess up a lot of folks if you don't do your part. So the team aspect is another big difference between the two environments.
I think it also depends a little bit on the company you are in. So if you're in a bigger company, it might be different to a smaller company. So I have worked now for 2 smaller companies, and I'm still doing research. And I think that's not that common in the industry, that you're really doing, you can really do your research. Normally you have a focus on what you have said, that you have a focus on one topic and you have to solve a problem. But sometimes if you are in a small company, you might have the possibility to do still research, and a little bit comparable then to the university research.
Any other? I think I saw more than one hand come up. Go ahead.
You guys probably had a lot of changes in career path over time, and my question basically is, how and when do you guys decide it's time to take a change or keep chugging on with what you're doing at the current time?
Okay, I'll restate the question. Is whenever your career has changed in the past, how did you know it, and how did you react, and were you happy with the result?
I guess because I took the biggest career change, I'll start. And it changes, it's variable. Sometimes you're forced to make a change, like Susan described. Other times, for me, I had this growing awareness that I wasn't very happy with the work I was doing. I didn't like the grind of writing grants in academic research, and I wanted to be more on a team again. And so that's it was that awareness that led me to make the decision to change careers. And I think in our culture we kind of have this expectation that people know what they want to do when they're very young, and that somehow you make that decision once and you do it the rest of your life. And I would just like to say, don't think that. You know, you never know what you want to do when you grow up. Even here, I think we're still thinking about what we're going to do next, and you know what we want to do next. So don't feel like you have to decide, you know, next year what you're going to do for the rest of your life.
I'd say, I guess, follow your gut. I know it's so wishy-washy to say, but I think, again, there may be a certain career path that you think you're expected to follow, but ultimately, again, follow what you're passionate about, follow your gut, and I think ultimately that makes you do better work because you care about it, and honestly, work doesn't feel like work when you are really excited about it, and so that's what's going to make you excel, and you know, and then is bound to result in that next step, which, you know, in my case, I don't know yet. I'm still earlier in my career, but so far, again, maybe I've started to take an unusual path, but I've followed my gut.
So I would say, just to echo something that Susan said, is you can't be bound by the fear of failure. So a lot of people keep doing what they're currently doing because they know how to do it. It's risk-free because they do it pretty reasonably well. But a risk-free life, doing something you don't necessarily like that much, is not very exciting. And it doesn't play to the passion that I think would, if you unleash it and take a risk, and you may even fail, but I think taking risk is much more desirable than just, you know, being satisfied with what the status quo is, whatever that is. I mean, you have to take risks. And that's what, you know, the Bay Area is known for entrepreneurs. There are entrepreneurs from all over the world that come to live in our environment just because they're kindled by it. So I would say that compared to students that are interested in science across many geographies in the country, you're advantaged because you're in an environment that understands what that's about and actually rewards it. So don't fear failure, and don't be afraid of the passion to try something different.
I must say, for me, it was a big step from coming from Germany to the US, so it wasn't my decision. So my husband came here for his employer, and I went with him. So I was really afraid if I can do that and if I can find a job here because I knew nobody. And but it was a great challenge. So I must say, to have another environment, to see how is it going in another country with different people, it's really a great chance to see something like that. So I think it's really worth also to go to another city, to go to another country maybe, and look how it works there. So.
Okay, are there any? Did you have?
Unless I'm obliged to do
No, you're not obliged to. There's no. I didn't see that on the. No, but any other questions? Go ahead.
If you could change something about your career choices that you had made in the past and you could do it in the career you're now, what would you change?
I might have gotten into toxicology just because it's really cool, but it's not because I don't like the chemistry, but toxicology is kind of like the coolest chemistry reactions going on in the liver and the, you know, in the gut and stuff like that. It's really cool, but that's just because I've learned more about it. I didn't know about that. You don't have chemistry, and you have chemistry in high school, but you don't have, you know, toxicology and things like that. So some more specialized fields like that you don't get exposed to. And I mean, I had a great experience. I had great mentors as an undergrad, and but I just didn't know about that field and didn't discover about it until I got into drug discovery because I was more on the metal, inorganic chemistry side of things. But toxicology was everything I went into.
So I think I wouldn't change anything because, yeah, I love what I do, and I also like to have the chemical background. So maybe I can, well, for me it's the right background, and the studies were great. So, but I also know now the biotechnology is very interesting. So no, I think I wouldn't change anything.
I never considered medical school really when I was doing my undergrad, unlike 99% of my colleagues. And that was because I wasn't really interested in treating patients, and I was interested in medicine in theory, I guess. But what I didn't know at the time is that there are a lot of MDs that don't treat patients, have careers not treating patients. And so it was only in grad school that I ever even considered potentially doing medical school. Again, I don't think it's held me back in my career so far, but I can't say it's speak too late, in the future down the road. So I guess that's the one decision that for now I wonder about.
I'd say the same thing as Stephanie. I wouldn't change anything. I've definitely made mistakes in my career, but I think as long as you learn from them, I think you're going to keep moving forward. Taking those risks, going off and starting a company, I've done that. I left a good job at one company, went off to start a company with another guy, and we moved into a garage, the classic startup tale. I built the lab benches, I did the plumbing, we cleaned the toilets, we did everything. We went out and we did that during the day and then put on suits and went out and tried to raise money. We failed, but, well, we raised some money, but we failed as a company. But I learned a lot from that. I think as long as you can learn from the experiences that you have, you're going to do fine.
Okay, go ahead.
I have a question again about career changes and possibly deliberating between two possible paths. It seems like when you go through a career and you go through your education, you start to specialize more and more, and you become an expert on that one specialty. So if you're really interested in a lot of different fields, how do you sort of combine those two while going into a specialty and still, maintaining connections with other areas of sciences that aren't really connected with your discipline?
So the question is, as you go on through your career path, you're often asked to choose to make choices in specialization. How do you maintain connection to all the different areas you might be interested in? Is that pretty much right? As somebody who's switched a lot from math and chemistry and genetics, immunology, gene therapy, I was all over the board. What's been the common connection for me was my basic love of science, the process of science. As long as you understand that the process of asking a question, designing the experiments, making the observations, and drawing conclusions from those, I think you can apply it to any field. When you do make a switch from one area of specialization to another, there's always a period of really intense learning where you are sort of behind a little bit, and you've got to put in some extra effort to catch up. There's no doubt about it. But I think learning a lot of different areas can really make you much stronger as a scientist. I think it's a good idea for your career to move around a little bit.
Yeah, I have a couple comments as it relates to that question. The first comment would be, increasingly institutions are recognizing how advances in science can no longer be, you can no longer be an inch wide and a mile deep. At least you can't do that by yourself. And so there are programs that are designed that deliberately force interactions between disciplines to affect success of individual projects. And so there's an institution like that at Stanford that encourages this. The University of Colorado, my alma mater, also is recognizing how important it is to have quantitative biologists together with molecular biologists, and actually in an academic institution work together in teams. Second comment would be as it relates to the team environment that takes place in companies. And so, like Alex said, that he really came to appreciate the finer points of toxicology probably after he got into a company environment. And that opened up a whole new window in terms of fertilizing his interest. And there's an iteration that takes place in a pharmaceutical company that the inputs on an individual scientist on a project team, you have to appreciate the scientific discipline, at least to an extent, of somebody who's around the table trying to advance a project. And so working in teams, whether it be in an academic environment or a company environment is incredibly exciting. And when you're in a company environment, my proviso or my advice to people is to stay off the critical path. So everybody's motivated not to be slowing the team down. And so working as a team it's incredibly motivating to get things done, and it can happen again in both the academic world as well as the industry world.
And a lot of those opportunities come up because you have a problem. So you learn about a specific field because you can't make your goal before you learn about an area of medicine or biology that you knew nothing about. So, you run into a drug-drug interaction situation, for example, then I have to really learn the ins and outs of that in order to get around it in my job. So that's a lot of those issues that come up that are problems for you. They end up being, what precipitates a crash course in a new area for you. And that happens all the time. And you don't know what they're going to be. I could run into that new problem tomorrow and go, oh God, I have to learn a whole new field. That could happen tomorrow. So that's part of what being a scientist is, kind of cool.
And I would add that, you have control of what you're interested in. And, to be a scientist at this time in human history, you do have to be specialized to a large extent. But you've got people around you who are doing different things, and also you have, more than one interest. You have another hobby. You do a double major. You keep yourself broad if that's where your passion is, and maybe you find something else to do in addition to science.
Okay, I think we have time for one more question, if there is one. Go ahead.
Basically, what I've heard right now is that, a lot of you, you worked in scenarios and where you've, formulated really, breakthrough projects and ideas, and I was kind of wondering how you formulated these breakthrough and revolutionary ideas and projects.
Okay, so the question is, first of all, thank you very much. I accept the compliment on behalf of my panelists. And I would say that every panelist here would love to think that they're actually working on a breakthrough project. And you have to accept that. But the question is, how did we get to work on exciting and breakthrough projects? Where did those projects emanate from? Where did they come from? That's the question, I believe.
Dave, why don't you answer that? given your history at Amgen.
So I would say a lot of it is fortuity. Scientific breakthrough is not exactly like engineering. It's having a background and a skill set and just working with great people. I worked on a project that began in 1994 with the discovery of the function of a novel protein we called OPG, and the long name is osteoprotogerin. And that was a novel discovery at the time that we ciphered through the use of transgenic mice that exhibited a change in their bones. And starting with that discovery, over a 15 to 16 year period, we identified the pathway in animals that was impinged on that, identified what the true drug target was, and then identified the drug candidate antibody for that, and then proceeded to develop it through a period of 5 to 7 years. And so I didn't make that mouse. I certainly helped make the observation and helped develop the biology. The mouse was made by a molecular biologist who was functionating novel genes, the biology of which was not at all understood. And he didn't know that he was going to be making a mouse with a bone phenotype, and I didn't know he was going to be making a mouse with a bone phenotype either. But together as a team, Bill Boyle and I, working with a lot of other people, made, in the mid to late 90s, a series of, seminal observations in an area of skeletal biology that was, presumed to exist, but it wasn't clear. So I would say you have to have an appropriate background. You have to be working with really great people that have personalities that are such that they don't have to own it all. It's not all about them. So you have to be in a very sharing environment, and then you have to be under the time clock that, hey, we're not here just to sort of do things for fun. We have a mission to develop whatever we discover into a therapy as quickly as we can, because drug discovery in America and throughout the world operates on an economic underpinning that makes it necessary that you are as thrifty as you can be and you make longitudinal progress. And so that sort of those constraints actually, for people that can respond to those constraints, actually yields a lot of creativity, because in my experience, creativity has not emerged during a time of plenty, but creativity is really the application of your mind and your resources in a very narrow way to get things done quickly, and that takes creativity.
If I could also offer an answer to that, I think at a fundamental level, breakthrough science comes from having an open mind and paying attention. You know, that it's easy to ignore a result that's not consistent with your hypothesis, especially if it's not entirely clear. You know, it's easy to kind of ignore that result and keep on the path you were planning anyway. And sometimes it's that unusual result or that unexpected result that's really the foundation of the breakthrough.
Yeah, I was going to speak to that as well. As an example, my PhD supervisor, I mentioned he was a virologist. He studied an insect virus called vesicular stomatitis virus throughout his PhD. And then like 20 years later in his career, he discovered that this virus is really good at infecting human tumor cells. And then he thinks back, he's like, well, you know what, we always grew that virus in human tumor cells back in the 70s. You know, it grew to very high titers. It was always very good at killing them, and they never just thought to make that link. And so, 20 years later, he's, made his career. So I think it is being the open-mindedness and thinking about potential applications or taking note of the unusual result.
I once saw, there's a pretty famous chemist who actually used to live in the Bay Area named Wilhelm Keim, and he used the term directed serendipity, which is obviously an oxymoron, but you really do have to keep in mind you have a goal at the end, but you also don't want to have blinders on, and it's a balance. And I think all of us have worked with people who have been too much one way or the other, and where somebody's just focused on, I'm going to get this, square peg into this round hole, and it's like, that's actually a really interesting square peg. Take a closer look at it, But he's just hammering the hell out of it, And so you find that a lot. But directed serendipity, I thought that was an absolutely brilliant way of putting it, because you do need to stay focused, but you still have to remain open-minded.
So is it my turn?
Yeah.
Anyway, I've been told by the program manager that it's time to move on to the next segment. I would like to take a moment for you to acknowledge our nice panel discussion. I hope you've gotten some value. I've gotten some value out of this discussion, so I hope you have too. So now we're going to move on to the award section. You don't need to stay up here any longer. We're going to go through the award section. I would just say I was one of the judges, and I had the fortunate responsibility to lead the discussion to arrive at identifying the finalists. And I can tell you that that's not an easy thing to do, because we looked at so much good science today. So I want to commend everybody before we start handing out the awards for all their good work. I know it's nerve-wracking to sort of come to a strange place and stand in front of 5 to 8 strangers that are listening to your work, but I have to, for everybody in the room that wasn't in there, I think you have every reason to be very proud of the performances of everybody in the room. They all did a great job, and I'm enthusiastic about the future of biotechnology in the Bay Area based on what I've seen today.